So let's talk about the probably the most exciting field in myeloma right now, which is immunotherapy, particularly active cellular immunotherapy, but BCMA as a target has emerged. Ed, your center has been at the forefront of CAR T therapy. Tell us a little bit about where are we at with CAR T therapy, and just let's start there.
Well, CAR T therapy has been the most exciting explosion on the radar of therapy.
Overhyped?
They'll realize that these studies really started of cellular immunotherapy are about 20 years old. So it seems like it's brand new, but it actually, is it overhyped? Well, there's no question that there are patients who've received these therapies who were dying of the disease that they were treated with and are now long-term doing well. So that's exciting and remarkable. Whether it is the cure for everyone with myeloma is still something that needs to be worked on. The way I see it is we have proof of principle. These therapies are definitely working, but now it's an engineering problem where we have to engineer these cells in a way that will be more broadly applicable and lead to long-term cures for patients.
So that's a good introduction. Yesterday at this meeting we heard some very specific data on one of the compounds from Bluebird and Celgene called BB2121. Dr. Chari, what do you think of that? I think maybe for the audience, just recap what we were shown.
So the BB2121 was a study with very heavily pretreated patients, median of 7 lines of prior therapy, and as Tom alluded to, I think up to 23 was the upper limit of the range. So in this heavily pretreated population, there was a very rapid response rate of nearly 100%. And I think what we learned new this time is really the PFS, which we really didn't have. And the PFS was around 11.7 months, I think. So I think what that tells us is, first, this is an amazing result for such a heavily pretreated population. That response rate and the PFS is superior to all other drugs that have typically been tested in this heavily pretreated population. I think the one caveat is the nitty-gritty of how patients get onto the study. So you need to meet progression criteria when you sign consent. But from that point to the administration of CAR T, the manufacturing process, it's anywhere from 4 to 6 weeks. And although you're allowed bridging therapy, who you're going to allow to sign consent is one selection bias. And then the second is who can actually make it to that 4 to 6 weeks is the second one. But putting that aside, undoubtedly very impressive results.
Tom, what do you think?
Yeah, no doubt the patients that were enrolled in this trial were the worst of the worst. They failed proteasome inhibitors, they failed IMiDs.
Their disease was the worst of the worst.
Yeah,
the
exactly.
They were wonderful people.
They were very wonderful, and failed daratumumab. And so these patients, we did not have real therapeutic options for them. And despite that, the response rate was tremendous. People say that the progression-free survival of 12 months, is it long enough? And they received the CAR T therapy, and then they received no other therapy, half of them for 12 months. If we could do that with other therapies, we would choose that therapy in a second to be a one-and-done type therapy for a year. I think this has dramatic implications, especially as in these end-line patients. So now we have to move it closer to front, early relapse, front line.
I think maybe there was a little bit of deflation with the PFS, because I think there had been this notion that, like the acute leukemia children, that this was going to be a one-shot cure, and that's probably not the case. If you treat people at this stage of disease, some will likely be cured, but there are others who are relapsing.
But I think that issue is also relating to the engineering that Ed mentioned, which is there was one neurotoxicity early, which subsequently has not been seen again with the lower dosing. So I think we need to really learn better about the dosing of the cells and the vector copies.
But I think it also, Harry gave a talk and said, he mentioned a patient, first CR, said, when can I get CAR T? To speak to the toxicity as well and the early experience in CAR T cells.